Welcome. Let's look at fertilization. Fertilization is the process by which the male and the female gamete come together to form the zygote. The coming together of the male and the female gametes is a very interesting process. Let's go through this lecture to see the basics behind the fertilization process. Fertilization, as we've said, involves the process by which the sperm cell and the egg come together to become the zygote. This is the egg, and these are the sperm cells around it, highlighted in blue. So it only takes one of this sperm cell to become fused with the egg to form the zygote. Let's look at the basics behind fertilization. We have the egg that is released by the ovary. At the point of release, this egg is also released with granulosa cells that are referred to as the corona radiata cells. If you look at this image, the structure highlighted in blue is the egg. And if you look around it, you will see that it is being surrounded by a group of cells that are referred to as the corona radiata cell. This cell tends to give a protection to the egg so that the egg is not just released on their own. So they tend to provide a form of guide or like an escort to the egg. So as they are released into the fallopian tube. This is the terminal end of the fallopian tube, and this is where the egg is sucked into, and it passes through the lumen of the fallopian tube until it gets to this region where fertilization will occur. At the point of ovulation, it is important for us to know that this egg is halted at the stage of the second meiotic division, which is the metaphase phase, so that after fertilization, it's going to resume the second meiotic division. So it's like an half-baked egg kind of that has not completed the second meiotic division, and this will be completed until when fertilization occurs. So it is not completely mature for fertilization to occur at the point at which it is released. So this is the part by which they run. So looking at the structural configuration of the sperm, this is the head region, and distal to the head region, we have the middle piece. Distal to the middle piece, we have the tail. So we have the head, the middle piece, and the tail. In the head, we have the nucleus, and this is the nucleus. On top of the nucleus, we have the acrosome. And this acrosome is very important in initiating fertilization process. We'll see this as we go through with this lecture, the role that the acrosome play during the process of fertilization. And more distally, we say we have the middle piece. The structure that is seen in the middle piece region are the mitochondria. The mitochondria help to produce energy in form of ATP, and this energy is being used up by the tail to exhibit whip-like movement so that it can be able to enhance the movement of the sperm, because they actually need to move through a path before they finally get to the point where fertilization will occur. So this is the configuration of the female reproductive tract. This is the uterus. Distal to the uterus, we have the cervix, and distal to the cervix, we have the vagina. And we know that vagina, of course, links to the external environment. So at the vagina, we have the insertion of the penis, and during ejaculation, the sperm is released, and when it is released, it is taken through the vaginal canal to the cervix. From there, it runs up and it passes through the uterine tube. This is the uterus, this is the uterine tube. The lateral extremities of the uterus is the uterine tube. So it passed through the uterine tube until when it gets to the specific region where fertilization will occur. So this is the path by which the sperm also runs. You can see that the sperm needs to run from one region to the other to the point where fertilization will occur. And also the egg that is released, we also need to run and move through to the point where fertilization needs to occur. So they need a form of movement, and this, of course, will be enhanced through the different structures seen within the female reproductive system. But this basic look very easy, but it's beyond what is presented. You have a lot of dramatic events that occur before fertilization will take place. So let's follow through to see the details of what will play off for fertilization to take place. So where specifically does fertilization occur? We've said that this is the uterus. On the lateral end of the uterus, we have a tubular extension called the fallopian tube or the uterine tube. That's the tube of the uterus. And this is the uterine tube. This uterine tube is further subdivided into three regions. We have the isthmus, This is the isthmus region. This is the region that is closely located to the uterus. Then more laterally, we have the ampulla, and this is the ampulla region, extending from this region down to this region. Then distal to the ampulla region, we have the infundibulum, and this is the infundibulum. The infundibulum is the terminal end of the fallopian tube. So out of these three regions, the region where fertilization takes place is usually in the ampulla region. This region, harrowed red, this region is the longest and the widest region of the fallopian tube. And it is in this region that the microenvironment is most suitable for fertilization to take place. Also, as the egg is released, it passes through the infundibulum to get to the ampulla. The egg does not have enough energy to drive through to the isthmus, so it stays here. Also, this can be used to justify the reason why fertilization occurs in the ampulla, because of the energy that is required for it to further move through to the isthmus. So it will rather remain in the ampulla region where fertilization, of course, can still occur. The region where fertilization occurs, specifically within the uterine tube, is in the ampulla region. Talking about movement, we said that the egg is released. As it is released at this point, there is no structural connection between the ovary and the terminal part of the uterine tube. They are closely located. The terminal end of the uterine tube presents a finger-like expression that is called the fimbriae. This fimbriae tends to exhibit a form of suctioning mechanism, so that as soon as the egg is released by the ovary, it's going to suck it up and drive it in through the lumen of the fallopian tube. So this is the basic of events that actually occur within this region. There is no connection between the terminal end of the uterus and the ovary, but they are just closely related to each other. And in order to prevent the falling off of the released egg, there's going to be a suctioning mechanism that is going to be set in place when ovulation occurs. Then another event that also enhances the movement of the egg along the tract, and also the sperm cell that is discharged or released into the vaginal canal to move to the region where fertilization will take place, is the muscular action. The muscular action, in terms of contraction of the uterus and the uterine tube, allows for the upper movement of the sperm cell and also the oocytes. They tend to contract, and as they contract, they help in propelling upward the movement of the sperm and also the egg. So the kind of contraction that they exhibit is an upward propelling action, so that the egg will be pushed and move from the infundibulum down to the ampulla, and also the sperm will be moved from the vagina to the cervix, then back into the isthmus before they finally arrive at the ampulla region. So it's an upward movement in helping to move these gametes. Also, to add that the epithelium lining the uterine tube also help to contribute to this movement. The uterine tube is lined by a simple columnar ciliated type of epithelium, and we know that ciliated epithelium tends to enhance movement. When the cilia flip forward and backward, they help to propel the substance or the structure that is located within the lumen. So because of the flipping action of the ciliated epithelium lining the uterine tube, is also going to enhance the movement of the egg to the point where fertilization will occur. As we already know that fertilization will occur in the ampulla region, and of course the release of the egg is around this region. So there's going to be a need for movement to be enhanced so that the egg will be able to reach the said region. We've talked about the regions where fertilization occurs, but before fertilization can occur, categorically, the sperm must undergo two reactions. So let's say this is the egg and this is the sperm that is coming to fuse with it. The sperm will not just come and fuse with the egg. It needs to undergo some form of reaction before they can have the capacity to fuse with the egg. So it's not just a direct coming together. It is not as it is presented. A lot of dramatic events will need to be unfolded before the sperm can actually fertilize the egg. So the first reaction that the sperm must undergo is capacitation. After capacitation, this will be followed up with acrosome reaction. So after undergoing this, then the sperm can now be able to fuse with the egg, and that fertilization would have occurred. So let's look through the two reactions that the sperm will need to pass through before they will be able to fertilize an egg. Capacitation basically is a functional maturation of the spermatozoa, which helps to enhance its fertilizing ability. And this basically involves the removal of the glycoprotein coats that overlies the acrosome. This is the acrosome, the region that is highlighted in blue, seen above the nucleus. This is the nucleus. So this coat covering the acrosome will need to be ruptured and removed so as to expose the acrosome. So this is the glycoprotein coat still intact in this sperm cell. And in this sperm cell, the glycoprotein coat is being removed. So this process of removal of the glycoprotein coat is called capacitation. What does this structural occurrence give to the sperm cell? So this allows the sperm cell to be hyperactive. As soon as this is removed, they become very active. So also more ATP is being produced and the flagella is able to flip more intensely. So they will be able to move more faster than the sperm cell that still has its glycoprotein intact. So we can say that capacitation will allow the cell to be hyperactive and also increase their motility. They will be able to move faster. It will also allow the spermatozoa to be able to cross through the coronary radiator cell. We said that when ovulation occurs, the egg is not just released. The egg is released with accompanying granulosa cells, which are called coronary radiator cells. So these are the coronary radiator cells highlighted in black. You can see them. Spermatozoa that undergo capacitation will be able to cross through or pass through the coronary radiator cell. So they will need to pass through this group of cells that surrounds the egg before they can finally reach the egg itself. And of course, it is after this process of being able to pass through the coronary radiator cell that they will now be able to undergo acrosome reaction. So let's see what acrosome reaction entails. But before then, the first phase of movement will then be the penetration of the coronary radiator cell. So the first passage part for the spermatozoa is for them to be able to have the capacity to pass through the coronary radiator cell. So for them to be able to pass through the coronary radiator cell, they must have undergone capacitation. Of course, they will need to pass through this cell before they can reach the egg itself. So this is like an initial phase of movement or penetration. And of course, this is enhanced by hyaluronidase. This is an enzyme that is contained also within the acrosome. When these enzymes are released, because the glycoprotein coat is already removed, so it will be easy for the acrosome to release hyaluronidase. So as it releases this enzyme will help to break up or digest the intercellular matrix between the coronary radiator cells. So it will be able to break it up, and as it is breaking it and digesting it, is able to pass through it. So this is the basic mechanism by which the capacitation enhances the movement of the sperm cell through the coronary radiator cells. And also flagellar action of the sperm cell is also important in this regard. We said that mitochondrium is located within the middle piece, and this mitochondrium tend to produce energy in form of ATP for the tail, so that the tail can have more strength to flip. And as it is flipping, it's also increasing the strength at which it is moving. So it can push harder to also force the penetration through the coronary radiator cell. We can see that is a collection of events that actually allows the penetration of the sperm cell through the coronary radiator cell. A number of events does occur before this can be allowed. So let's go to the acrosomal reaction. This is a reaction that follows with capacitation. We now know that the sperm cell now have gained entrance through the coronary radiator cell and also at the point of penetrating into the egg. But before they penetrate the egg, they need to undergo acrosomal reaction. So what does this involve? This is an exocytotic form of reaction whereby enzymes are released by exocytosis under the induction of calcium inflow. So as they approach through the egg, it releases the acrosome. And this acrosome, we already know that is a cap-like structure that is located on top of the nucleus. It is already exposed because the glycoprotein coat is removed. So as it gets here, it releases the acrosome, and the acrosome will help to digest the zonal pellucida. The zonal pellucida is the region highlighted in red. This is like a tough coat that surrounds the egg. It may also be referred to as the shell of the egg. So it will digest it, and after digesting it, it will be able to penetrate into the cytoplasm of the egg. So the first phase is to pass through the coronal radiator cell. The second phase will then be to pass through the zonal pellucida. And at this stage, we said that fertilization has occurred, but there's still more to this. At this time, the sperm cell will be able to fuse with the plasma membrane of the oocyte. So at this stage, we said that fertilization has occurred, but there's still more events that will structurally and functionally stage that fertilization has occurred. Facts that we need to know, we know that during ejaculation, about 200 to 300 million of sperm cells are released, but 300 to 500 of them will reach the point of fertilization. That is the ampullar region of the fallopian tube. And also to add that out of these 300 or 500, only one of the sperm cell will be able to fertilize the egg. After one lucky spermatozoa is able to penetrate into the egg, the other ones around will degenerate. We cannot allow more than one sperm cell to penetrate into the cytoplasm of the egg because it will create some abnormalities. So we say that after fertilization is now completed, we cannot structurally or functionally assume that fertilization has taken place. Some processes also will need to occur. And this is how the egg responds to the contact of its cytoplasm with the spermatozoa. 3 processes are also initiated for us to be able to say completely that fertilization has occurred. The first one is zonal reaction. After the zonal reaction, we have the resumption of the second meiotic division of the egg. Then the third one is metabolic activation of the egg and the sperm to form the zygote. So the zygote will need to be formed before we say that fertilization has occurred. Because while defining fertilization, we said that it's the coming together of the male and the female gametes to form a single zygote. So let's see how the female egg also responds to the contact that it now has with sperm cell. So for zonal reaction, the zonal reaction basically entails the blocking of the zonal pellucida. We already said that the zonal pellucida is a tough membrane that surrounds the egg, and this one lucky sperm cell is able to cross through it. This highlighted in yellow, where we have the other white ones surrounding it are unable to cross through. This, of course, will undergo degeneration. For this, after entering, there's going to be the initiation of zonal reaction. The zonal reaction involves the alteration in the structural composition of the zonal pellucida so that there will be blocking of the zonal pellucida to prevent the inflow or the invasion of additional spermatozoa. So this tends to prevent polyspermy. We already said that if more than one spermatozoa fertilizes the egg, it will lead to abnormality. So in order to avoid that, there's going to be the blockage of this region highlighted in green, which is termed the zonal pellucida, so that other spermatozoa will not be able to penetrate through. And also more specifically, there are sperm receptors located on the wall of the zonal pellucida. These receptors are thus destroyed so that the sperm will not be able to fit into any receptor before they can now have access to penetrate, because there is no structure that is binding them onto the wall of the zonal pellucida. They would have been destroyed. Also in a way to prevent polyspermy. There's also the resumption of the second meiotic division. Remember, we said during ovulation, the egg is not completely mature because it is halted in the second meiotic division, specifically in the metaphase phase of the second meiotic division. So it is after the contact between the sperm cell and its plasma membrane that they are able to initiate the completion of the second meiotic division. So there's going to be the production of the mature egg now and also another polar body. Then the third activity is the activation of the egg and the sperm cell to form zygote. We said that the sperm cell is made up of the head, the middle piece, and the tail. The tail is degenerated so that it is only the nucleus that is introduced into the cytoplasm of the egg. The tail is to enhance movement for fertilization to occur, and it has exhibited its own property, and that will mark its end. So it's going to be degenerated, while the nucleus will be released into the cytoplasm of the egg. And as they are released, the male and the female pronuclei will be seen in the cytoplasm of the oocyte. And if you look at it, they are not differentiated. They're almost looking like the same at this point. Then the next phase is that there's going to be the degeneration of nuclear envelope, so that the two pronuclei can fuse together to become one. And we know that the male and the female pronuclei, they are made up of 23 chromosomes each. That is haploid number of chromosomes. So we have the two haploid coming together to form the diploid number of chromosomes. And the diploid number of chromosomes is what is expected in the normal developmental form. The fact that the sperm is able to penetrate the zonal pellucida, thereby having contact with the plasma membrane of the egg, is not enough for us to say that fertilization has completed, until when there is blockage of polyspermy and also resumption of the second meiotic division and activity that will involve the coming together, the fusing together of the male and the female pronuclei to form the zygote. So all these processes also will need to occur before we can completely say that fertilization has occurred. So going through the consequences, generally, the consequences of fertilization would be that it will stimulate the secondary oocyte to complete its second meiotic division. And we know that this is initiated after the contact of the sperm cell with the plasma membrane of the egg. This is able to stimulate this occurrence. And the restoration of the haploid number of chromosome into the normal diploid number of chromosome. We know that the male and the female pronuclei each has 23 number of chromosomes, which will come together to form the normal chromosome. So this is one of the consequences of fertilization. Also, variation of human species. We know that the genetic component of both parents will be integrated and intermingled, and that's what gives the variation of human species. And also the determination of sex is achieved during fertilization, and this is determined by the male factor, whether they donate the X or the Y chromosome. If the X chromosome is donated, then it's going to be a female child. When the Y chromosome is donated, there's going to be the production of a male child. So it depends on the limb that is donated during fertilization that will determine the sex of the baby. Then lastly, there's going to be the initiation of cleavage. Cleavage is the next phase that will occur after fertilization is said to have completely occurred. So cleavage will not occur except if fertilization has occurred. That is what it means, and this is one of the consequences. And when cleavage occurs, there's going to be mitotic division of the cell. Then this will progressively lead to the development of the different structures and organs in the body. Clinical anatomy. We said that more than one sperm cell cannot fertilize the egg. There are a lot of mechanisms that are put in place to check this, but there are instances when this may occur. This appears to be very rare. So you have one that claims that it is the first, while the other one feels that it's not the first. So at the end, this is going to create some form of abnormality, as we've said. This is going to cause polyspermy, which involves the fertilization of an egg with more than one sperm cell. And we already said that we have 23 number of chromosomes in the oocyte, and this will merge with the 23 number of chromosomes in the male sex cell, which is the sperm cell, to form the normal diploid number of chromosome, which is 46. So when we have more than one sperm cell, this is going to alter the configuration of the genetic component or constituent of the zygote that will be formed. There's going to be the formation of an abnormal set of chromosomes. This situation is very rare, and when it does happen, the embryo that results from this usually dies off with time because they do not have the capacity or the grace to live beyond the stipulated time, because the configuration that they constitute or they form cannot be sustainable. So another instance is the formation of both identical and unidentical twins. Identical twins are also referred to as monozygotic twins, which means that they exist from one zygote. So there's a formation of a single zygote that we already know that is formed as a result of one sperm cell fertilizing and egg to form the zygote. So this zygote then divides into two, and this is where we have the formation of identical twins. So the twins that is produced from this kind of formation, they tend to be genetically identical because they are formed from a single zygote that is formed as a result of the fusion of one egg and also one sperm. It is just that after the formation of the zygote, they tend to divide, thereby having the same genetic component, forming the twins that would tend to be identical. Then we can have the formation of unidentical twins, which is also referred to as dizygotic twins, which means that they exist from two zygotes. And this is formed when there's a release of two eggs. Two eggs is released, and each of the individual eggs is fertilized. So you have two separate formation of zygote from different egg and also different sperm, and they tend to develop together, and that will result into the formation of unidentical twins. So that is the basis onto which the identical and non-identical twins are formed during the process of fertilization. So thanks for watching this video. If you do enjoy this video, kindly inform others of it so that they can also benefit.